Hello everyone, welcome back to another session of RNA biology. And we were here in the previous class and we were discussing about the nuclear export and nuclear import of various RNA cargo into and out of the nucleus. So the mRNAs, they follow a different strategy, different track, unlike rest of the RNA, which depends on the RAN GTPase cycle. So we saw in detail the RAN GTP and RAN GDP have got gradient across the nucleus with a purpose that it will be helpful in mobilizing various helpful or supportive proteins for the movement of RNA cargo in and out of the nucleus. Now let us see a diagrammatic representation of the nucleoporins in the RNA export. This slide will look very complex and quite cumbersome, but understand this many number of proteins are present in the nuclear pore complex and there is no easier way we can simplify it. If you want to know the strength and depth of these proteins, we should understand there are mainly three different categories of proteins in the nuclear pore complex. The ones which provide structural support of the nuclear pore complex, which do not play any role in the transport of the cargo, that is the RNA cargo, but they just hold the nuclear pore complex into the membrane. So nuclear membrane is a bi-wall structure, two-wall structure is there and they are present, and remember it is a three-dimensional structure. What you are seeing is a cross section and it is like a pipe, it is just like a cylinder. Now you have some proteins hanging into the cytoplasm and there are some factors hanging into the nucleus, and left hand side you can see typical metazoan, that is all multicellular organism. Right hand side you are seeing yeast specific proteins, that means you have NUP84 complex whereas NUP107 complex in the metazoan. So this is the main difference. So functionally they are similar, but name wise or amino acid sequence wise they are different. So same way for rest of the proteins. So the entire half you can see, and you can also see many FG proteins hanging into the lumen, and you saw in the previous class that the FG proteins are the pockets. We gave the example of rock climbing, etc., the pockets in which the cargo literally interacts with them. And so you can also see many associated protein along with the structural protein that is present for interacting with the cargo, or the proteins associated with the cargo. That is, here cargo we are referring to the RNA species. So this is the nucleoporins in the RNA export, mainly in the metazoan and also in the yeast. Now let us see in detail with proper description how each of these proteins are important in the transport of the cargo. So nucleoporins in RNA export. So nuclear pore complex NPC is formed by approximately 30 different nuclear pore proteins, and we call them as nucleoporins, that exist in 8 or 16 or sometimes even 32 copies per nuclear pore complex. Not the point that they are in pairs. There will not be 9 or there will not be 17. So they are 8, 16 and 32. So they are kind of occupy the diagonals quite effectively. So depending upon which cell type or which cell's nucleus you are talking about, accordingly, like some tissue there will be active transcription going on, like say liver or sometimes neuronal tissues, etc. So they need to have more effective, more efficient migration. So they may have more proteins associated, like less transcription is occurring like in some of the epithelial cell or some of the lining of the intestine, those kind of cells, They don't have any dedicated function. They are playing more of a structural role. So the gene expression events may not be that robust as you see in the case of some of the glands or some nervous tissue like that. So nucleoporins are grouped into three major classes. The first class are the FG nucleoporins, that is those nucleoporins that contain the FG repeat, phenylalanine glycine repeat, which contain this phenylalanine glycine rich repeat domains that fill up the active transport channel and function directly in the nucleocytoplasmic transport by mediating the passage of the soluble transport receptors 161 and 162. So these are the FG nucleoporins. What is the another class? They are the first class of nucleoporins. The second class of nucleoporins that are devoid of the FG repeat sequence. They do not contain the FG repeat. These are the predominant structural constituents of the NPC. That means it provides structural support. The nuclear pore complex should be of a particular shape and they need to have this structural support. The third class are the NUPs, which are integral membrane proteins that are thought to anchor the nuclear pore complex to the nuclear membrane, because they are also structural in category, but they have nothing to do with the nuclear pore complex functioning. They are holding, they are acting like anchors or hooks, just like if you are hanging a fan in the ceiling, you need to have a metallic hook, and metallic hook has nothing to do with the functioning of the fan, except that the fan is in place hanging from the roof because of this hook. So something like that. This is the third class of protein. So let us see. The first class are the FG nucleoporins, and the second class are the nucleoporins that do not have the FG repeat, and the third class are the NUPs. They are basically holding the nuclear pore complex onto the nuclear membrane. So the RNA export pathways, you can see them in this cartoon, that the RNA category include transfer RNA, microRNA, snRNA, mRNA and ribosomal RNA. And the movement of all these RNA category, like tRNA require RAN-GTP cycle, miRNA require RAN-GTP cycle, snRNA require RAN-GTP cycle, and mRNA do not require RAN-GTP cycle, ribosomal RNA require RAN-GTP cycle. You can also see the associated protein, like here exportin T, whereas here it is exportin 5, and here you can see a CRM1, and like that each of these molecules have a different set of associated proteins, and these associated proteins help in two ways. One thing is recognizing the nature of the cargo, whether it is a tRNA, whether it is an snRNA, whether it is a microRNA. This recognition is done by these proteins. And another way is that they have to have a specific downstream event. That means presence of a given protein associated with the cargo can decide what should be the destination of this cargo. It's not that they are just associated. How effectively this should be transported, what is the rate of transport, everything can be decided by the protein associated with that. So we should understand this cargo protein complex, depending upon which species being transported, they will have a dedicated exportin associated with that, and this is the nuclear pore complex, and what you see downwards is the cytoplasm. So based on the nature of the RNP that is formed or assembled, accordingly it will pass through the nuclear pore complex. And nuclear pore complex proteins also interact with specific proteins associated with this RNP. So let us see individually how different RNA species are transported. Do they have anything in common or are they unique? Let us see transfer RNA and microRNA processing and their export through the nuclear pore complex. Let us see tRNA export at first. It has the same principle of exportin mediated protein export, what we saw earlier, that the tRNA export receptor, that is exportin T, it is a member of the karyofurin superfamily. It binds directly to the tRNA in a RAN-GTP dependent manner. RAN-GTP, we will not go into the details. We know that RAN-GTP, it provides the phosphate to the karyofurin for the release of the cargo in the cytoplasm. Otherwise, in the cytoplasm also it will hold the cargo, and we don't want that. So the nuclear export signals in the tRNAs are decoded by exportin T. So this decoding is very important, because without the exportin T, the tRNA's nuclear export signals cannot be read. So exportin T's role is to identify the right cargo. So the NES, nuclear export signals, are not just a linear motif, not that U, C, A, U, like that you saw poly A signal, which is A, U, A. So it's not like that. That's a linear motif, but here it is not like that, but they are present in the form of secondary and tertiary structural elements at the 5' and 3' termini. So this is what you should understand, that they have a unique dedicated secondary structure that is obvious in the 5' and 3' terminus of this tRNA molecule. Once the tRNA exportin and T-RAN-GTP complex reaches the cytoplasm, the RAN gap, that GTPase activating protein, RAN gap stimulates the GTP hydrolysis on the RAN to induce the release of the tRNA cargo. Otherwise, exportin will not release the cargo. It will hold on to the cargo. So the moment the RAN GTPase is acted upon or activated by RAN gap, then you will have the release of the cargo. Another exportin 5, is a member of the karyofurin family, and it can act as a auxiliary receptor. That means it is kind of a supportive in function. Sometimes in some tissue, if the exportin T is not available or abundant, then, or exportin T is not functional adequate enough, then the auxiliary receptor, it's just like an assistant, just like a personal assistant. You have personal assistant cannot replace you, but personal assistant can enhance your efficiency. So this is exportin 5 can act as a auxiliary receptor. An alternate pathway of tRNA export. Let us see, is this the only way? What you saw in the RAN-GTPase exportin T mediated, is there any other method? And in yeast, the tRNA export receptor LOS1 is an ortholog of exportin T. Exportin T is not there in the yeast, whereas you have another protein called LOS1. But interestingly, it is not essential for viability. It may sound little surprise, because you cannot assume that the tRNA need not be transporting to and from the nucleus in the case of yeast. That's not the case, because LOS1 is the functional counterpart of exportin T seen in metazoan. So how does this happen? The function of LOS1 tRNA export receptor became essential when the activity of an amino acid tRNA synthesis cofactor known as ARC1 was impaired. So what it indicates, LOS1 is doing the job of exportin T, but if LOS1 is missing, then it is not affected. So we should assume the tRNA transport is not affected. So we should assume there is some assistant, someone else is doing the function of the LOS1, and we could guess it, who could be that be. So if LOS1 is defective along with the ARC1, which is a amino acid tRNA synthetase cofactor, then absence of LOS1 become important. So another protein, that is SEX1, was suggested to deliver the aminoacylated tRNAs from the nuclear export receptor at the cytoplasmic side of the nuclear pore complex. So another protein, SEX1, was able to facilitate the movement of the or the maturation of the tRNA even in the absence of LOS1. So ARC1, if it is missing along with the loss one, it becomes problematic because another protein Sex1 was acting as a mediator for the interaction of the tRNA with the amino acid tRNA synthetase. So we should understand systems or the organisms, they evolve with diverse strategy. They can have unique strategies through which they evolve. And this you also may remember that we talked about the evolution by gene duplication. So this can be one example. Like loss one gene is important, it is necessary, it is doing its job, everything is working fine. But there is also a second in charge or second person in charge which is there, a second molecule in charge is there. So ARC1 and Sex1 can work together and they can complement the absence of loss one when it comes to the transport. So naturally what happens in evolution, the loss one can acquire some novel functions eventually. So whether it acquire or not is a question of time, but it is possible because loss one is kind of free. Even if it is present, it is kind of free from doing this job alone. It can do another job. So this is an example which we can think about, that ARC1 and Sex1, they can kind of complement the absence of loss one. But if ARC1 also is missing, then it is a serious issue. So you can read this review, that is exporting RNA from the nucleus to the cytoplasm, which came Nature Reviews Molecular Cell Biology a few years ago, and it's quite interesting, and I have used some of the images from this review, and it's an excellent review. So students should go through it for a better understanding of the subject. So tRNA processing and export, like you can see in this picture, a tRNA normally is transcribed with RNA polymerase 3, and it has to undergo the maturation process, and once it undergoes maturation, then this 5' and 3' processing and the CCA addition of the tail, everything has to happen. Now in the nucleus you can have three species. One is tRNA with intron, that is immature, and it can have the CCA maturation already, and or properly processed tRNA, and you can also have amino acylated tRNA. So all these three has to find its way out. It is holding the tRNA along with the intron, probably because it's defective. It has to be marked for degradation, and some of them the amino acylation will happen in the cytoplasm, not in the nucleus. For some, the amino acylation can happen in the nucleus itself. So for all this category, you need to have exportin T and exportin S, and also other pathway proteins. They have to come together and act along with the nuclear pore complex proteins, and they move into the cytoplasm. And in the cytoplasm, it will disassemble because the RAN GTPase cycle come into picture, and the RAN gap will help in the GTP hydrolysis, and the cargo will be released and the cycle will continue. The amino acylation can happen to those tRNA which do not have the amino acid attached. The amino acylated tRNA can directly participate in the protein synthesis. Remember, there are 20 different amino acids, and you also have 30 different tRNA species which has to be catering the 61 different codons of the genetic code. And let us see how the biogenesis and export of microRNAs happen. MicroRNAs are group of non-coding RNAs similar to ribosomal RNA and transfer RNA, but their role is post-transcriptional gene silencing. That means they are performing the gene regulation which is on the mRNA after the transcription. So miRNAs are a class of non-coding RNA. They are exported by the karyofurin exportin 5. We saw for tRNA it was exportin T. So miRNAs are produced as larger stem loop precursors in the nucleus which mature into single-stranded RNA species that induce post transcriptional gene silencing, PTGS. Transcriptional gene silencing means a gene regulation occurring at the transcriptional level. That means whether or not a RNA is produced or not. Whereas post-transcriptional gene silencing means it has to be regulated after the fact or after the production of the mRNA that is happening in the cytoplasm. MicroRNAs encoding genes can be transcribed by either pole 2 or pole 3. So they can be depending upon, like we will see them in detail, because microRNA can be embedded inside the intron of an mRNA. So in that situation, the microRNA transcription is regulated by polymerase 2, RNA polymerase 2. Whereas some microRNA can have their own promoter, their own genes. So that transcription is polymerase 3. So the primary transcript derived from polymerase 2, we refer to them as miRNA, that is primary microRNA, and it transiently receives a 5' cap and a poly A tail because that is embedded inside the intron of some other gene. The cap and the poly A tail are removed during subsequent microRNA processing. Many a times this microRNA, if they are transcribed, they even can have their own 5' and cap and the 3' tail, which will be removed, because eventually the microRNA are just 22 nucleotide in length and they are functioning in the cytoplasm onto the mRNA target to prevent their translation. So let us see the biogenesis and export of microRNA in detail. The stem loop structure that is present when it is formed, it harbors a 22 nucleotide microRNA motif. Remember, the stem loop structure of the microRNA can be around 150-160 nucleotide. So somewhere in this 150-160 nucleotide you have a complementary, means both strands are pairing each other. That is why it is able to form a stem in the first place. Loop region doesn't have any pairing. The stem region have got complementary sequence. That is why it is forming a stem, and it has around a 22 nucleotide long microRNA motif, which is excised and exported and finally assembled into the RNA interference effector complex. So we call it as RNA induced silencing complex, or it's called RISC. The stem loop is cut out in the nucleus by an RNase 3 enzyme, and we call it as drosha in the complex with another protein DGCR8. So remember, the stem loop structured microRNA is acted upon by RNases, drosha, along with the help of a protein DGCR8. So stem loop can be of around 65 nucleotide in length. The 65 nucleotide stem loop intermediate, we call it as pre-microRNA, is exported. Earlier it was pri-microRNA, and now after cut by the RNase 3, drosha and DGCR8, we call this product as pre-microRNA, which is now around 65 nucleotide stem loop intermediate, is exported to the cytoplasm in a RAN-GTP dependent manner by exportin 5. So exportin 5 is contributing to the transport, and the GTP hydrolysis in RAN that results in the release of the cargo, that is the 65 nucleotide stem loop intermediate microRNA into the cytoplasm. So the pre-microRNA hairpin is further cleaved by dicer. So this action happens in the cytoplasm. So another nucleus called dicer comes into picture, and this produces, so this is also an RNase 3 family protein, which produces a 22 nucleotide microRNA duplex. So this is precise. No matter which microRNA you are talking about, it is 22 nucleotide in length, which is in complementary sequence, it is paired. And one of the strand is the one which participate in the silencing event. Both strands do not participate. So one of the strands of this 22 nucleotide participate in the silencing event. Now in this microRNA biogenesis, you can see in this cartoon, you have this nucleus and you have this primary microRNA which has formed the loop, and this is from the DNA. The primary microRNA has formed, and this is the stem loop structure which is acted upon by Drosha nuclease and DGCR8, and you end up getting a pre-microRNA, and this pre-microRNA is now, with the help of exportin through the nuclear pore complex, it is released into the cytoplasm. So in the cytoplasm, the exportin is released back because of the RAN-GTPase cycle, and this pre-microRNA is now acted upon by dicer along with another protein FMRP, and you end up getting a microRNA duplex which is 22 nucleotides in length, and this can assemble into RNA induced silencing complex, RISC complex, onto mRNA species, and this complex involves a bunch of protein. One of the major protein is argonaute protein, which binds onto the mRNA and causes the degradation, and it can cause translational repression, that is preventing the mRNA from protein being translated, or mRNA cleavage. The mRNA can be marked for degradation. Both way it functions. So we can see more in detail about the microRNA and its function in the next class. Thank you.